Welcome to saitechinfo channel, my dear students. In this tutorial, I'll be explaining about proteins, that is the chemistry of proteins. We will be discussing amino acids, then classification of amino acids, followed by structure of protein, and I'll be explaining about the denaturation of protein. We have already explained that the proteins are the polymers of polypeptides, and polypeptides are the combinations of peptides, and the peptide that is resulted due to the combination of amino acids. So the fundamental unit is amino acid. What is amino acid? We know very well acetic acid, CH3COOH, and this is called alpha carbon atom. So in place of alpha hydrogen, you can introduce amino group. So that forms NH2CH2COOH. And this is known as amino acid or glycine. This glycine, see, we have got variety, almost 20 types of amino acids. In glycine, we will try to arrive at some general formula so that it would be easy for you to understand the remaining set of amino acids. For instance, so here we have CH2, and let me put here R, and this is COOH. So let me replace one hydrogen with amino group. So this is the standard structure. So when R is replaced by hydrogen, which is nothing but HCHNH2COOH. So this is known as glycine, and its one letter code is G, and three letter symbol is Gly. Similarly, alanine, so you can replace this R by CH3, then you will get alpha amino propionic acid, which is nothing but alanine. So when you replace this H by CH3 twice, so what do you call this? This is isopropyl group, is it not? So just by replacing this R by isopropyl, you have CH and then CH3. So this part is called the isopropyl group. So isopropyl group, and that is known as valine. So it is represented by V. Alanine by A. And alanine can also be represented by three letter symbol Ala. Similarly, valine, that is VAL. And what about this leucine? So what is leucine? You just count the substitution, the number of carbon atoms in the substitution. 1, 2, 3, 4. That is butyl group. But how it is arranged? This is an isobutyl group. Are you able to understand? There are 4 carbon atoms. It is arranged, let me draw here, that is CH3, C, CH3. So this is what here it is represented as CH3 twice. Okay, and then CH, right? Then CH2. So this group is introduced in place of R. Now you can continue with the remaining part, that is CH2, NH2, COOH. That's all. So this is the R part. So based on the R part, we are able to get different types of amino acids. Do you understand this? Right. And then, so you just count, I think that is, I should put only CH here because it will affect the tetravalency. Okay. So CH. Now let us consider that is, replace this R with H. Okay, H, and this is also H. So there are two H and one amino group and COOH group. Whereas here there is one hydrogen, one carboxylic acid, one amino group, one isopropyl group. So this is not an asymmetric carbon atom. So it is not optically active. Not optically active. Whereas the remaining amino acids are optically active because this carbon atom is attached to different atoms or group of atoms. For example, in the case of valine, it is attached to isopropyl group, amino group, COOH group, and then H. So all are different, and that is why it is having asymmetric carbon atom. So they are optically active. So except glycine, all are optically active because it results in asymmetric carbon atom. So this is a very important point you have to note down. Now, by introducing this isobutyl, you are able to get leucine, which is nothing but L. It is simply represented by L. So likewise, we can see the remaining 16 types of amino acids, like isoleucine, and then arginine, lysine, glutamic acid, aspartic acid, glutamine, and then asparagine, then threonine, serine, cysteine, methionine, and phenylalanine. So phenylalanine, that can also be illustrated here. We know already alanine. What is alanine? Let me try the structure. C, and this is H. So this is NH2, and this is COOH. Now when you put CH3, this is alanine. Now it is phenylalanine. So it means you have to substitute with phenyl group. So CH2, C6H5. I hope now you should be able to understand how to write down the structure. So phenylalanine, that is represented as Phe 3-letter, and F, that represents the single one-letter code for phenylalanine. Then we have triosine, tryptophan, histidine, proline. So these are the different types of almost 20 amino acids are available, among which, as I already mentioned, all amino acids are optically active except glycine. So that point should be noted down. Now let me look into the classification of amino acids. See, that is, when you look at the structure, you have both amino group and COOH group. So a molecule that has amino group, that represents the basic nature, and this is the acidic nature. So it means it can have, that is acidic nature, basic nature. But this hydrogen, this can be transferred, the proton transfer can take place within the molecule. So this H plus, that is, it gets removed as H plus and it's joined in the NH2 group. So it becomes ammonium here. So this is the basic radical that you know very well. And what about this? This is minus. So plus and minus, they exist together. So whether it should be called acid or base, in this case it is neutral, and that is why it is called Zwitter ion. So it is an ion that consists both cation and anion in the same molecule. So generally amino acids are colorless, they are crystalline solids, they are water soluble, high melting, and as we already mentioned, it has COOH and amino group, acid group, carboxylic acid group, as well as amino group. Zwitter ion is a very popular ion. In fact, see, that is when this amino acid solution is subjected to electrolysis, we can anticipate the anions. Which is the anion? For instance, RCHCOO, I can write like this, NH3, NH2, CH, you can put R here, then COO minus. This is anion. What about the cation? That is NH3 plus CH or COOH. This is the cation. And which is the Zwitter ion? That is dipolar ion. It has got both ion. This is known as Zwitter ion because it has got both. Now one important point is here, I mean it is to be studied here, is the movement of these cation and movement of anion. This we know during the electrolytic process. But when you have the formation of Zwitter ion, to which direction this ion will move? It will not move. Why? It has got both positive and negative. So there is no current because of the formation of this Zwitter ion, and that point is called isoelectric point. Isoelectric point. Are you able to follow this? Isoelectric point. It means during this electrolytic process, there is no movement of any ion because of the formation of this Zwitter ion. Okay, if this cation and anion move, towards which direction they will move? This plus ion, cation, will move towards cathode. And what about this? This anion, they move towards anode. But what about this? It will not move. At that point is called isoelectric point. Then, and we have already discussed about the optical activity because of the presence of asymmetric carbon atom, but in glycine there is no asymmetric carbon atom. So that is why except glycine all are optically active. Then naturally occurring amino acids, they have L configuration, that is this NH2 is written on the left hand side. Now let us move to the next one, the structure of proteins. Proteins, the polymer of alpha amino acids. For instance, let me consider NH2CH2COOH. This is one amino acid part, and let me consider another one. So in this case, this is amino acid, that is glycine. This is glycine. And what about this? This is alanine, is it not? This is alanine. They can combine together to undergo a condensation, that is, there will be a removal of water, that is OH and H will be removed, so that you are able to get this CONH. This CONH bond, that is the linking between, that is the linkage between the two amino acids. It can be of same type or different type. So this is called peptide linkage or peptide bond. So And this is called a dipeptide because it forms out of two monomers. So glyala, or you can call it GA. Do you understand this? Right. So likewise, you can have 3 moles or 4 different or 4 similar. Either way, it can be combined to result tripeptide, tetrapeptide, pentapeptide, then polypeptide. So in this case, I have shown a dipeptide, that is glyala. So when the polypeptides is exceeding 10,000 units, then it can be called protein. But we cannot take it for granted. Even with the 51 amino acids, if a protein has a specific biological function, then it can be called that is protein. That is polypeptide can be called protein. The best example is insulin, which has 51 amino acids. Now, what are all the various types of proteins? We have fibrous proteins, globular proteins, but basically there are 4 different levels of structure of protein. They are primary, secondary, tertiary, and quaternary. But the 3-dimensionally speaking, basically it has 2 important structure, that is fibrous protein and globular protein. Fibrous proteins, they are made up of chains. They are held by hydrogen bond. They are parallel, and they are linked by disulfide bonds. Then they are insoluble in water. The best examples can be keratin, which are available in hair and wool. Myosin, they are present in muscles. Globular proteins, that is spherical, globular, its structures are globular. They are soluble in water. Example, insulin, albumin. As I already mentioned, there are 4 different types of different levels of structures. We can call it primary structure, secondary structure, tertiary structure, followed by quaternary structure. Let me talk about this primary structure. So as I already mentioned, it is backbone of the protein. So the primary structure, it may contain 1 or more polypeptide chains, but it has some specific sequence of amino acids. So it will be like this. Then secondary structure, the long peptide polypeptide with the alpha helix. What is that? Alpha helix and beta pleated sheet structure, that is sheet-like type structure. Due to the regular folding of a backbone of a polypeptide chain, they can form, and it is because of hydrogen bond and the CONH peptide bond, and that is why you are able to get this alpha helix. Alpha helix means they are twisted, and they are right-handed twisted. So that is why they are called helix. So the structure is given here. This is the helix structure. Okay, the twisted structure. And then we have the secondary structure, that is a beta structure, sheet structure. So beta pleated structure. It is because of the cross linking. So they are stretched out sidewise, and they can be formed by the intermolecular hydrogen bonding. So they result such type of structures. And then we have the tertiary structure of protein. This is the overall folding of polypeptide chains, and the secondary structures can be further folded. Then it forms a fibrous and globular proteins that we have already discussed. So the forces of attraction among these structures can be, that is hydrogen bonds, disulfide linkage, van der Waals physical force, that is intermolecular force. Then it can be a Coulombic electrostatic force of attraction also. Now quaternary structure. So it can be 2 or more polypeptide chain. There will be some spatial arrangement between the subunits. Now let me display the overall structures, that is the 4 types of structure. So the primary structure is a linear one, that is the backbone. Secondary structure may be a twisted, okay, and tertiary structure like this, and the quaternary structure will be like this. In fact, each colored ball can be representing an amino acid. And this is further complicated one. This is the structure of hemoglobin. Hem means iron. Globin, globular protein. There will be 4 myoglobin. So this is myoglobin, 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 myoglobin. 4 myoglobin are combined together with the central metal atom iron. So where they are called hemoglobin, and that is why it's called hemo. Hemo means iron. So globular protein 4 myo. That is why the hemoglobin can be called the tetramer of myoglobin with the central metal atom iron. Now let me talk about this primary structure. So this is the primary structure. You know something about the joining of different amino acids. That results in the secondary structure that we have already discussed as the what? The double helix structures. Okay. And further, this is the tertiary structure. So it can form a tertiary structure where the R is represented here as a green ball, and this is heme is the iron atom. Okay, so likewise this is the final 3D structure, that is the quaternary structure. So for we have discussed about the structure of protein. Now let us get into the denaturation of protein. Nature. Denature means that is losing the nature of protein. For instance, native proteins are 3D proteins with unique biological activity and structure. But these hydrogen bonded structures may be disturbed due to physical change like temperature or chemical change like pH. For instance, if any acid is poured on our skin, that will denaturate your skin protein, is it not? And if the skin is subjected to some hot temperature, what happens? There is also the denaturation of. Denaturation means once for all it is irreversibly it is changing its nature. So the unfolding of globules and helix, then there will be an uncoiling of protein, so you cannot get it back, and it will lose its biological activity. As I already mentioned, every protein has got unique activity with its own structure, so it has lost here. And the denaturation is nothing but the loss of original nature and its shape. So the 2D and the 3D secondary structures are destroyed, but primary structures are intact, they remain unaffected. And the best examples can be coagulation of egg white or on boiling, then curdling of bacteria resulting lactic acid. So these are all the very good examples to explain about the denaturation of proteins. So I hope you should have enjoyed this lecture. Please go through all those points. The terms are very, very important, and try to develop concept map and finally arrive at a sketch based on the protein. Okay, so thank you very much for watching this video.